Okay, yeah, it's recording it. All right, so students, welcome back to the lectures on molecular motors. And what we're going to do today is we're going to look at another kind of molecular motor called kinesins. We have so far looked at actin, we've looked at microtubules, and we've seen how myosin motors function. So today it's the turn of kinesins. So, before I start the lecture, I'm going to play this video, and what you're going to see in this video are small microscopic beads that have been put inside a cell. So you could micro-inject beads inside a cell, like you could micro-inject DNA inside a cell. So here, scientists did that. This has been done a long time back. And then they found that the beads are actually moving inside the cell. So let's have a look at the video first. So you see the beads, they seem to undergo some sort of motion. And this, at least, you can see that it seems to be moving in a straight line, and you can actually see that line. And so this was kind of an artificial illustration of how materials within a cell is transported. Because inside a cell, now it's very simple to think of, say, an epithelial cell, or say the skin, the cells of the retina, the small cells. But think of neurons, which have their cell body somewhere in your spinal cord, but the axon has moved down your leg and is sensing through your feet. Now that the distance that that cell has to cover, right from the spine to your feet, that's an enormous distance, and the cell needs to transport materials through these long distances. So the question was, how is the cell doing this? How is the cell able to transport materials? And when we talk about materials, what are the things that we think that a cell may have to transport? It's not just different kinds of proteins or even sometimes mRNAs. It has been shown that cells try to preferentially keep certain mRNAs in a certain location where those proteins are required. For example, if a cell is migrating and the cell needs to make some proteins that are necessary for it to migrate, it so happens that the tips of those cells contain those mRNAs, contain those certain mRNAs that are instantaneously producing the proteins for the cells to move. Which indicates that there are a lot of things that are being transported, right? There are proteins being transported, mRNA being transported. Then there are organelles, because such a long cell would require ATP, so you would need transport of organelles, transport of vesicles. So all of these needs to be transported, and it's long-range transport we are talking about. So how is this accomplished? This was the question people were after. And so initially, because myosins were known, so they thought, okay, maybe myosin is what is causing such long distance transport. Myosin is the motor doing such a function. And then it turned out that myosins were in fact not able to answer this question of what was actually transporting these beads, because this was kind of a system, how do I call it? This was kind of recapitulating what is actually happening inside the cell. You put in beads and you see that the beads are moving, but the beads never moved with myosins, which indicated that there must be something else working behind this. So So this is, you can see this is a paper from 1985, much before you were born. Uh, and what they tried to understand is, no, they were trying to find out what exactly is causing the beads to move, but they didn't hit upon the molecule instantaneously. They first identified a few features of this transport. For example, if you look here, you would see that this is a bead, and with time, you see the bead has moved. So here is one microtubule, here's another microtubule, and they could clearly make out that the bead was moving on the microtubules, right? And then it was taking a few seconds to move from this microtubule to this microtubule. So they found that this movement was occurring in a manner where the microtubules were guiding the movement. And the movement could be done with these beads. They could also be done with organelles. For example, here, if you see here, with the arrow they have marked out, here there is an organelle, it's here at one time point, and at the second time point it's here. So how did they do this experiment? What they did was they took microtubules and then they added these beads or organelles, whatever it is, along with, they took axons, squid axons. Squids are known to have one of the longest axons in our living system. And so if there is something in this squid axon, if there is something in this squid neuron that is helping it to transport materials across long distances, then they should be able to figure out what it is, what kind of motor it is from that squid axon. So thinking of it in that sense, they simplified the whole thing. Instead of seeing it inside the cell, they took cell-free extract. Cell-free extract, there's another thing with the cell-free extract, that is, it would have all the proteins required, but it would not be the entire cell. So you're trying to kind of narrow down to see what exactly is causing the movement. Okay. So initially, what was it? They took a cell, put the beads inside, and then they saw, okay, the beads are moving in a specific way, in a specific orientation, a specific direction inside the cell. Now they took microtubules, they took the beads, okay, and they took a cell-free extract. That is, you break open the cell, you don't have the cell anymore, but you have all the proteins that were present inside the cell. So you take that, you add that to your mixture of the beads, and now you put that on the slide with microtubules attached to it. And what do you see? You see the movement of the beads. So there's something in that cell-free extract that is causing this movement. There's some motor there. They also, so as I said, initially they didn't hit upon exactly the motor, but they hit upon characteristics of this motor. And one of the characteristics was to deal with the ATP hydrolysis. So what did they do? They used this. This is adenylyl imidodiphosphate, AMP. If you look at the structure of this, you'd see that it resembles very closely the structure of ATP. So it's an ATP analog, but it's an analog that cannot be hydrolyzed. So ATP can be hydrolyzed to ADP, an inorganic phosphate, but this cannot be hydrolyzed. So what they found was that in the cell-free extract, when they were adding the beads, they could see the movement of the beads on the microtubules. But if they added this non-hydrolyzable analog of ATP, the movement stopped. So as you can see here, the moment they add this, there is no movement. It's all flat, dash, dash, dash. No movement. Similarly, if they inhibited ATPase activity, now they have the cell-free extract, they've added ATP to it, but if they inhibit ATPase activity by increasing the concentration of this molecule called vanadate, then again they can, so at 20 micromolars, not much was happening, but at 100 micromolars, when they increased the concentration of vanillate further, that's when they found that the movement of the beads slowed down or was completely absent in some cases, which indicated that whatever was the motor protein, it required hydrolysis of ATP to sort of help it in translocating this bead along the microtubule, right? Initial ideas when they did not really know what was causing this whole phenomena. All right, so then the task was very simple. You have seen that there is something in this cell-free extract that is causing the beads to move. Now the point is to find out from the cell-free extract which protein is causing it. That's a non-trivial question to answer, because as you can understand, a cell-free extract will contain thousands of proteins, right? So how do you hit upon that single motor protein that is causing it? So again, they went back to try and understand, using whatever was known so far, which molecule, which protein is doing it. So one of the first things they noticed was that when you have microtubules and you have these beads moving on the microtubules with the cell-free extract, the beads and the microtubules had to be, cell-free extract had to be, if it was pre-treated for a very short time with this non-hydrolyzable ATP analog, it increased the binding of the beads to the microtubule. But of course, because it was non-hydrolyzable, there was no movement of the beads, but it increased the binding. So then the authors thought, okay, it might be that something like myosins, because myosins was known at that time when they were doing this work. They thought, okay, something like myosins might be happening here. So you have a condition where it is bound, but it is not translocating. And maybe that might be enough for us to isolate the translocator, or to isolate the motor protein. And that is what they did. Here is the paper that first went on to show kinesins, and the first paper that actually named kinesin. So here you can see is one lane of SDS page, which shows the presence of microtubules. So they took microtubules, right? Then they took the cell-free extract, you call that S2. The axonal from the squid axons, they took the cell-free extract, that's S2, right? You mix it in the presence of this non-hydrolyzable ATP analog so that the bead would not move. That's the condition. The bead would not move, but the bead would be bound to microtubule. And now you try to pellet the whole system. You pellet the microtubules and try to see what is binding to the microtubule. There must be something binding to the microtubule, because if it has to use microtubule as a track, definitely it needs to sit on the microtubule. And when they did that, this is what they found. They found a weak band somewhere around 110 kilodaltons, somewhere around here. Very weak band. So they went on doing this further, enriching this fraction over and over and over again until they could clearly see this band coming up, 110 kilodalton. And they also observed a dependency on ATP. So as they kept increasing the concentration of ATP, they could get more and more of this translocator bound to the microtubule fraction. So they could get it released from the microtubule fraction, more of it released from the microtubule fraction, as they kept increasing ATP concentrations, which indicated that this particular 110 kilodalton protein is actually the motor. Now, to finally prove it, they had to take this 110 kilodalton protein, isolated, then put it on the cover slip with microtubules and beads and ATP, and show indeed that the bead is moving. So that's the final experiment to conclusively prove that 110 kilodalton protein is actually what is causing movement of the bead. But this itself went on to show that they were on the right track, that this 110 kilodalton protein has a dependency on ATP when it comes to binding to microtubules. Okay, so what are the caveats that we have seen or that they saw in this experiment so far? The translocator does not bind tightly to microtubules. One, no microtubule movement occurred when microtubules were treated with the cell-free extract, centrifuged, washed, and resuspended in an ATP-containing buffer. So that's the first thing that they noticed. As I said, they could understand that the to actually have uh, to actually have this bead binding strongly to the microtubules, the first thing that they needed was to have a non-hydrolyzable ATP analog that could help these beads attach to the microtubules in the first place. If not, this event was not very stable. So this binding of the translocator, that they call them translocator, or the protein kinesin that we know now, was not very was not was the binding to the microtubules was not very tight, but it did occur. And so for them to work with the cell-free extract and with the beads, the first thing they needed to do was to use this non-hydrolyzable analog, which induced the attachment of the bead organelles, whatever it is, to the microtubules. Okay, addition of this non-hydrolyzable ATP analog to purified microtubules and the cell-free extract increase the amount of polypeptide that co-pelleted with microtubules. So again, here, so the more they add, so first is that if you have ATP, this event is quite transient in nature if you're seeing this in a cell-free extract. In a cell-free extract, always remember this. What they're seeing here is a cell-free extract, not exactly the condition in a cell, but a cell-free extract. Okay, so but if you add a non-hydrolyzable analog of ATP, you can make the bead stay on the microtubule track, and that you'll be able to visualize its presence on the track. But having the non-hydrolyzable analog is not going to move the bead. To move the bead, you have to add ATP. And if the ATP removes the non-hydrolyzable analog from the kinesin molecule, then you'll be able to see not just the binding of the bead to the microtubule, but also its translocation. With that, let us now come to see what is a kinesin molecule. So now we have seen that for myosins, the myosins were walking on actin tracks, right? Were walking on actin tracks, and myosins were powered by ATP. Does somebody remember the cycle of how myosins were utilizing ATP and then binding the power stroke?
Yes, ma'am.
Yeah, go ahead, please.
So, ma'am, what happens when myosin binds to ATP? The affinity for actin binding will be reduced. So, when it will be in ATP state, it will be free from actin binding. And when it will get hydrolyzed, this will regain the activity to bind again to this actin filament, and when PI is released, then it will lead to a power stroke that will lead to the movement of myosin and pulling or pushing of the actin filament.
Thank you. Thank you. Perfect. Perfect. So you see, we are now going to understand kinesins by taking an analogy from myosin. Because again, as I said, this whole idea of motor proteins, how they function, is all about two things. One is the hydrolysis of ATP or GTP. And the second thing is the hydrolysis is coupled to a conformational change. So if you have things bound to ATP, that's in one conformation. ATP hydrolyzed to ADP, but the inorganic phosphate is still bound is another conformation. Inorganic phosphate released, bound by ADP, another conformation, right? And for kinesins, let's say removal of ADP completely, no ATP or ATP bound to it is another conformation. So you have this series of changes of conformation, all being powered by hydrolysis of ATP. So that was happening in myosin. The same thing is happening in kinesins. In kinesins too, you have the motor being powered and driven by ATP hydrolysis. But it works in a slightly different fashion, and we're going to come to that. So kinesins, first of all, they use microtubules to move. Like myosins were using actin to move and were literally kind of sliding the actin filaments and sometimes even walking on actin filaments. Kinesins, they walk on microtubule rails. And kinesins, as we have seen, they are required to transport materials inside the cell. And kinesins are not, there's not a one gene for it, but there are about 45 mammalian genes for kinesins. So in our bodies, we would have about 45 genes which would code for this kinesin superfamily. That's what they're called, kinesin superfamily, because there are just so many kinds of kinesins. Of course, each of these genes could give rise to multiple forms of the protein because of alternate splicing. So the diversity of kinesins is enormous. But kinesins can still be grouped into a certain subclasses. And one of the way you can group the subclasses is to see where exactly the motor domain of this kinesin molecule is. We'll look at this figure here, and you can see that it says N kinesin, M kinesin, and C kinesin. So N for N terminus. So you have a polypeptide chain and you have an amino terminus for the polypeptide chain and a carboxy terminus. Nish, if you have difficulty understanding, let me know. So you have an amino terminus, you have a carboxy terminus of a polypeptide chain. So an N terminus is the amino terminus. C terminus is the carboxy terminus of the polypeptide chain. Depending on where the motor is residing, it's at the N terminus, the N-type kinesins. C terminus, C-type kinesins. Neither N nor C, somewhere in middle, M-type kinesins. Very straightforward classification, and nothing big here. So you can see this green cylinder that has been depicted here shows you where exactly the motor domain is. So it's a huge family of superfamily of proteins, motor proteins, and there are multiple genes for it, 45 or so. And those 45 genes can give rise to even further diversity by alternate splicing. You can have a huge diversity in kinesins, but then some of these kinesins can have the motor domain at the N terminus, some can have it at the C terminus, some can have it at the middle. And in general, this is within quotes. In general, the N-type kinesins are the classical kinesins, the conventional kinesins that move on the microtubule towards the plus end of the microtubule. whereas the C-type move towards the minus end. And it's seen that the M-type kinesins are often there to depolymerize microtubules. So one extra level of information here, you can have kinesins that are moving towards the plus end of microtubules. You could have kinesins that move towards the minus end of microtubules. And we've already seen some kinesins that were helping in bending of the protofilaments. You remember we had seen an electron microscopic a reconstruction of electron microscopy images where it was shown that binding of kinesin to these protofilaments was actually causing bending of the protofilament. So you can also have kinesins that are helping in depolymerizing microtubules. So this is a kind of cartoonic depiction of what or how people see kinesins and their function relative to the microtubule. So here is a microtubule track, a rail track of microtubules with a minus end and a plus end. And the plus end is where new tubulin dimers are being added preferentially compared to the minus end. Of course, in cells, the minus end is always blocked because the minus end is attached to the MTOC. So the minus end is not actually free, right, in cells. Okay, so here you see this, you can have these, imagine this to be kind of like a wagon that's moving on a rail, and the wheels of the wagon are what are the motors. So you can have N-type motors, N-type kinesins, and those would be moving towards the plus end of the microtubule. You can have C-type that would be moving towards the minus end. You could have some which are M-type and which are actually involved in buckling, bending of microtubules, helping them depolymerize. And at the same time, these kinesins have to have the capacity to move things, right? That's the very first reason why kinesins were found out, because they were trying to find the motor protein that is required to transport materials across the cell. So then they should have the ability to hold cargo. They should have the ability to bind to cargo and pull it along. So then there has to be another part of this kinesin molecule that is binding to cargo, that is carrying the cargo, that is pulling along the cargo. So you think of it as a wagon where you have the motors that are binding to microtubules, helping in movement of the kinesin molecule. But at the same time, it is like a wagon and it is holding the cargo. But how exactly does this all come together? And how does this kinesin actually look? We'll come to it in a bit. But let's take an example of let's come to the structure first, otherwise I'll be having difficulty explaining. So this is a simplified cartoonic representation of a dimeric kinesin. Not to mean that a kinesin always acts as a dimer. Unlike tubulins, which are dimeric, you have alpha beta tubulin dimers, which are the basic repeat units. Unlike them, kinesins are not always dimeric. They could also be monomeric. But conventional kinesin, just to understand the structure, many of these conventional kinesins actually work as dimers. And even they could even multipolarize. So what exactly is in the structure? Let's go back. Yeah. You see, there is a motor domain, right? And then you have something called a coiled coil domain. And then you have this long stretch of amino acid, and then you have a pH domain. So this here is the motor domain. This is what is helping it bind to microtubules and walk on microtubules. And this is the coiled coil domain, which helps in formation of this dimeric structure. because the coil can coil on to another coil, and that gives rise to the stable dimeric structure. And here you have those pH domains which can bind to cargo. Now, binding to cargo could be direct through this domain, or sometimes you could have some light chains attached here, and those could bind to the cargo, or you could have some adapter proteins binding here, and those could bind to the cargo. So there are multiple ways by which the kinesin molecule can bind to cargo. And kinesins can also select the kind of cargo that they would carry. And it all depends on how this cargo is getting attached to the kinesins. So for example, some kinesins might require this light chain to bind to the cargo. So additional two chains can bind to the kinesin molecule, and those have an affinity to say certain mRNA, and therefore that can become the cargo. In other cases, you could have an adapter protein that comes in between, and then that can bind to an organelle, say mitochondria, and therefore, in this way, the kinesin can then transport this mitochondria on a microtubule tract. So there are various ways by which, or it could be a direct connection. There might not be a light chain, or there might not be an adapter protein, and the C terminus can itself bind to the cargo and can carry the cargo. So multiple ways by which kinesins can do it. Not very well, not very clearly understood how this huge diversity is achieved by kinesins, but more or less there are indications that if it can change the light chains attached to it, if it can change the adapter proteins attached to it, it can change the way or the type of cargo that it is carrying. Okay, all right. So another important thing is that this motor domain that you see here is also the site that binds to ATP, just like myosin. So in myosin, you had the motor head that had one point where it could bind to ATP and hydrolyze ATP, and diametrically opposite was a point where it could bind to actin, but everything was on the head domain. Similarly, here you have the ATP binding, ATP hydrolysis activity, all residing in this motor head. Everything residing in the motor head. Okay, now after this motor head, you have a region called the neck linker region. So this is the neck linker region. And what people found was that the orientation of this neck linker region with respect to the head, that changes whether you have ATP bound to the head, to this motor domain, or you have hydrolysis of ATP to ADP. So every time the ATP is bound to the head, this neck linker region seemed to bury itself inside the head. And that created a condition of the power stroke. So it is kind of a little different from myosins in the sense that here addition of and binding of ATP to this head is what is creating enough space for this neck linker region to bury itself. It just from being almost perpendicular to this head, it's now buried inside the head. So that's the amount of change in the positioning of the head with respect to the neck linker that occurs. And it's easier to understand if you look at the cartoon, but before that, let me just quickly summarize what we know about the cycle by which the kinesins walk. So let us consider a situation where you have this dimeric kinesin. As I said, kinesins, a single unit of kinesin can still function. It's not necessary that it has to dimerize. Many of the kinesins do work as dimers. So let's take a condition of a dimeric kinesin. And so we look at these two motor heads, and we think of them as the two hands that are moving on microtubules. And let us consider that one of that motor domain no longer has an ATP bound to it, and the other has an ADP bound to it. The moment ATP binds to this empty region, that's when, as I said, the linker sort of buries itself, the neck linker kind of buries itself inside the motor domain. And that propels this domain forward. And you would see that in this video. You would see that in this video. So here is the kinesin, the two legs of kinesin. So you have ATP here, and you see the neck linker sort of buries itself. That kind of propels this other molecule or this other head in front of it. So every time the ATP binds, the neck linker buries itself, and that causes a conformational change. It sort of kind of moves this molecule in a way that the other motor domain then falls in front and binds to microtubule. So the whole power stroke here is kind of driven by the binding of ATP to this empty pocket, empty nucleotide binding pocket of the motor head in a kinesin molecule. Okay, so far, am I going too fast? Is that okay so far? Hello?
Yes, yes.
Okay, all right. So as I said, thing here is for us to understand that there is a subtle difference between the way the myosins were working and the way the kinesin is working. The working principle is the same. You have ATP hydrolysis driving some conformational changes that is propelling these motors forward. But here is a situation where binding of ATP is actually producing a large change in the conformation. So again, I'm going back to this image because this is a very what should I say? This is a very straightforward way to understand what exactly I mean by conformational change and how much of a conformational change. So you see, what is happening is at the region where this linker, neck linker region is sort of attached to the rest of this kinesin head, you have a groove, but the groove is restricted, and therefore the groove cannot the groove cannot accommodate this entire neck link. But binding of ATP opens up that groove so that the neck linker region can bury itself inside it. So the whole this whole thing proceeds in a manner that binding of ATP causes a change in this groove region, opens up the groove a little. This opening of the groove allows the neck and the neck linker region to bury itself inside it. In the end, what happens is that you have a change. So the moment the neck linker region buries itself, this whole, because it's, you have to think of it, this whole thing is one unit, right? You have one conformational change, and that is sort of flipping the other foot forward. You have a conformational change here, you have a burying of the linker, and that changes the conformation of the whole molecule such that it is flipping the other foot forward. Okay, so now it has flipped the other foot forward. So the motor was here, right? And the other foot was here. ATP bound, the neck linker region buried itself, and then this propelled the other foot forward. But the other foot had ADP bound to it. So now that ADP is released, and when that ADP is released, you have this situation where the molecule is under tremendous force, because you have the neck linker region still buried inside this motor domain. So this is released by ATP hydrolysis. You have ATP which is hydrolyzed. The neck linker region is again back to its own place. And so it again comes back to this situation where it is waiting for this empty head to now bind to ATP and again flip this region because the neck linker region will bury. So the whole dynamism of this molecule arises because of ATP binding, and the ATP binding would sort of open up a small groove, and that would be enough for the neck and the neck linker region to bury itself inside that motor domain, propelling the other foot forward. Look at this video once more, very carefully, and you will see that the same thing is happening here. So here is the ATP that is binding to it. You see the neck linker region sort of buries itself. That propels the other foot forward. Okay, that propels the other foot forward. But where the other foot is going to bind to the microtubule is still sort of determined by Brownian ratchet. And this molecule is still sensing the microtubule around it to find its perfect place to bind. And it so happens that the perfect place for it to bind is the next closest dimer of tubulin. So you have this alpha beta alpha beta alpha beta spaced around or on the microtubules, right? You have the alpha beta dimers of microtubules. You also have the alpha beta dimers around it, so that because it's a cylindrical structure of about 30 protofilaments. So you have these alpha beta dimers, and so once one of the motor head is bound to one of the dimer, the other motor head would search for the next closest dimer. So the stepping size turns out to be exact match as the distance between these dimers. It's about 8 nanometers. Okay, so this is, let's look at it once more. You have ADP released, ATP binds to it, again change, linker buries itself, flips the molecule in front. This molecule searches out for the nearest dimer, binds there, another ATP, flipping again, and so on. Okay, I've missed some points so far. No, I think I'm all right. Now let's come to this. So with this, I'm kind of going to stop the class today. And this is where we're going to look at the stepping size of kinesin motors. Now, in the last class, I had discussed about the optical trapping and optical tweezers. So, what was? Can somebody just give a brief about optical tweezers, just as a recap?
Ma'am, in optical tweezers, it was like utilizing the light so that a bead can locate itself with any range of light, any particular wavelength. So when we will move that light back and forth, then that particular bead will also move within its vicinity of whatever the wavelength it was preferably having.
Yeah, I'm sorry I could not get your voice very clearly. Uh, so, um, can you repeat that once more, Farman?
Yes, ma'am. So what happens in optical tweezers? The beads are having a particular wavelength. it will stay basically uh, stats on a particular wavelength in a light when we split it. So when this light will move, we will also move in response to their wavelength. Wherever it will go, then they will move towards direction only, and so the movement will happen.
Okay.
So for that, I mean,
Okay. Okay. Thank you very much. So, as the name suggests, optical trap, it's a method to trap objects, microscopic objects, beads, cells, anything using light, optical trap. And tweezers, because just like you hold things, you know, like to tweeze apart, you hold things. So it's a way by which you can actually hold molecules. Um, so it's that's why it's an optical tweezer. So what is it doing? It's utilizing the property of certain properties of light. For example, the property of scattering, light scattering. So if light hits a bead, then there is scattering from that, so from the surface of the bead. But then the scattering is also going to change the momentum of the bead. And if you think of the way the when you have an intense laser spot uh, focused through lenses, you would see that the laser spot does not have uniform intensity throughout its cross section. You'll see that in the center the intensity is very high, and then it falls exponentially at the sides, which means that there is a distribution of intensity of light. So if the bead itself would see different momentum at different places depending on where it is on this light spot. And coupled to it is the fact that the light itself is an electromagnetic radiation, and the beads themselves are dielectric. So they also have some interaction with this electromagnetic radiation. And therefore, the whole thing together helps to trap these beads. So if you leave the beads under the light and you bring the light very close to the beads, you'll see that the beads are simply pulled towards the center and they're kept in place. Of course, there's a little bit of that motion, a little bit of that Brownian motion, but it's more or less kept in place in the center of that trap. So if you can trap a bead to the center, now think of it this way, if you can trap a bead to the center, now what if you attach your microtubule or actin or myosin or even kinesin to this bead, then every time the kinesin tries to walk, it would try to pull the bead along with it. But the bead is being kept in place because of this light, right? The light is trapping the bead, the light is keeping it in place. So it will not allow the bead to be pulled. But the myosin would still exert some force and would try to move the bead away, right? Because it's walking, right? So it would still try to move the bead away until it sort of fails and then it stops and then the bead would just go back. So what you can now the second thing is that it comes from Hooke's law, which says that the force applied on, so you think of a spring, and you think of a metal ball at the end of a spring. Now you move the metal ball for a distance of x, that would require a certain force. If you are to move the distance to 2x, you require twice the force, which means that the force is directly proportional to this displacement, right? F is directly proportional to this displacement x, which means that if you translate that to this system of optical trap, if you know that if my kinesin motor is walking on microtubule and it's pulling the bead along with it, with every step it's moving the bead, and if I know the displacement, I can calculate kitna force is पे काम कर रहा है. What is the force acting on this bead? So with this, we come back um, to this AFM measurement of a kinesin, two-headed kinesin motor. And what do we see here? So we see here called the bead displacement, and we see kinesin displacement. So bead displacement is going to give us. Uh, so let's now imagine what the setup would be like. So the setup would be something very simple. You would have a microtubule, and you have microtubule on a glass slide. So you take a glass slide and you're able to etch microtubules on the glass slide. You're able to put the microtubules on the glass slide. Now you have a kinesin motor, and you have uh, uh, you have been able to add the bead to this end of the kinesin motor, the end that usually binds to the cargo, such that the motor end is now free and it can bind to microtubule and walk on microtubule. But the end where it binds to cargo is now attached to the bead. Simple setup. So you have a bead, you have a kinesin motor attached to the bead, and the kinesin motor, the cargo end is attached to the bead. The other end is free and it can walk on the microtubule trap. Now, with the help of the optical trap, you have trapped that bead under an intense laser light. So you have trapped the bead. Now you add ATP, and this ATP would start to would sort of allow this kinesin motor to walk on the microtubule. So every time it walks on the microtubule, it puts that step, it moves another step. So it tries to pull the bead along. So you would be able to see the bead displacement. And if you can also measure kinesin displacement by some means, say you've tagged the kinesin with GFP, and by some means you're able to sort of measure that from time point X to time point Y, what is the displacement of the kinesin itself. So you can have two readings. So let's forget one of the readings. Let's think of the bead itself. So suppose you're able to track the bead as it moves, because the kinesin is moving on the microtubule. It's trying to pull the bead along with it. It's trying to pull the bead along with it. And you're able to see how much of the distance the bead has moved. And then after some time, you would see that that it can no longer move the bead, because the bead is going to go back into the trap. It's going to just fall back into the trap. So here is a situation where you can see that this bead, we are looking at the displacement of the bead. So you see that the bead, it's moving about its mean position in the trap, and then the kinesin starts walking, and then you see it moves back again. Again there is a movement. Again there is a movement. So you see a whole range of these movements that it takes place before the bead goes back into the trap, which shows that there are a few things that you see here. First is that kinesins can move without falling off the microtubule processively for a considerable distance. So it's called processivity. How long an enzyme, say a DNA polymerase or an RNA polymerase, can be bound to DNA or RNA and can produce new DNA or new RNA without falling off the molecule. So that's processivity, right? So the PCR, in PCR reactions, the thermus aquaticus, Taq polymerase, whatever you use, has a certain processivity. But that processivity is not enough if you would like to, say, amplify 10 kilobase pairs or 15 kilobase pairs. You need something with more processivity. So you change the enzyme then. So what is processivity? Processivity is the point that how far these motors can work without falling off the strands. So here the kinesin, for kinesin, it would be how far it can walk on microtubules without stalling and falling off the microtubule. So the processivity of kinesins is very high. it can keep working on microtubule without falling off it and without stalling, without stopping. So it can walk a considerable distance. So that's the first thing you see here. You see that the force is building up. So it's walking. The bead displacement is occurring. You see the bead. It's displaced. It's displaced. It's displaced. It's displaced until it falls back into the trap. So there is a considerable amount of processivity in the way the kinesin motors function. Second thing you notice is that the step size, here the step sizes, they have indicated it with these dash. To me, it was not very clear, but just when, if they hadn't added these dashes, it would have been very difficult for me to even explain this. But if you look at each of these steps, so you have one step here, second step here, third step here, fourth step here. So each of these steps, so every time there is a slight movement of the bead, and then another pull at the bead, another pull at the bead. So that's the trace lines that you see here. And between these, each pull and each movement of the bead, if you measure the distance that it has moved, it turns out to be about 8.2 nanometers. That's the spacing between adjacent microtubule dimers, which indicates that the kinesin motor is actually seeking out its nearest microtubule dimer when it is walking on microtubule protofilaments. Okay. And then you can also calculate, by looking at the displacement, you can calculate the force, and it turns out to be of the order of some piconewtons. Okay. Kinesins take many steps, processivity. All right, so with this, I would stop today's class. But if you have any questions, I can please go ahead. I would like to answer them.
Ma'am, I had a question. Uh, Errors while walking on this microtubule.
Huh? Microtubule?
Uh, I'm saying like when kinesin walk on this microtubule, they don't have like it sometimes like errors. Like if it have if error happens, then it can fall apart from this microtubes microtubules. If this
Uh,
thing happens.
well, you see the reason why they are able to walk on the microtubule is because of the tight coupling with ATP hydrolysis. That actually creates a state where it is very stably bound on microtubules. What we are looking at here is a condition where we are simulating it with a bead that is actually in a trap. So ये condition थोड़ा सा different है. In cells you will see that they walk for so microtubules can form very long tracts in cells, very long tracts. And these kinesins can actually walk on these long tracks, very long tracks. They have very high processivity, so they will not, in general, they will not fall, but they can stall. What is stalling? So stalling is where they can stop in the middle of the track. You can so when we deal with dynein, after that I'll show you a video. So you could have a situation where you have a traffic jam kind of a situation. You have various motors that are trying to walk on the microtubule, some walking from the plus end to the minus, some walking from minus to the plus, and they can meet at a point and they can stall. It would take time for them to resolve कि कैसे हम, how are we going to manage going past each other, considering that both of them will be carrying cargo. So stalling can occur. Uh, Falling off is not that that what should I say? I shouldn't have said falling off. Processivity ऐसे हम सोचते हैं falling off, but microtubules के ऊपर ये fall off उतना नहीं कर सकता because it's so tightly coupled to this ATP hydrolysis. And to the way that it walks, it's energy dependent. So it is So the interactions and everything are tightly coupled to this ATP hydrolysis. So it's more about, I should say, stalling, or yeah, something like that. But it's not that it might not fall off. Of course, there is always there's always a K off. Like in chemical reactions, there's always a K off. There is always something, some constant, rate constant associated with a with a kinesin that would fall off a microtubule. But in reality, this becomes highly processive because it is coupled to ATP hydrolysis. That helps this molecule to always keep its one foot on the microtubule and walk on the microtubule. Oh, do you have any other questions? If not, we'll carry on the class. Next Tuesday, we'll carry on with kinesins, and then we will move on to dining, and then you have your minor exams. So something for the minor exam. Your minor exam is going to be open book. So you do not have to bother about memorizing a lot of things. What I would like you to do is to understand the concepts, because your minor would be an open book exam, and it would not be a multiple choice exam. So you're not going to be ticking the correct answers. Be prepared to write on that day. You'll be asked to write or to envision certain situations, or to explain a certain uh, experiment or design an experiment kind of a thing. So study whatever material you have so far, look at the class lectures that I have taken, and you will be able to answer the questions. All right, students, so we'll see each other on Tuesday, and we'll continue with kinesins. Thank you so much.
Ma'am,
Thank you.
like
Okay, ma'am. Thank you so much.
how much marks will be like for minor?
Huh?
Like what will be the weightage of minor?
Oh, the weightage of minor is 40. 40 is the minor, 50 is for major, and 10 would be for a project that I will give you after the minor.
Okay, ma'am.
Okay? Yeah.
So the project will be like will have to like some term paper or something, or
It'll be
like
it'll be a short project. It'll be a short project for 10 minutes.
okay, ma'am.
Ma'am,
Thank you, ma'am.
minor exam will be on Moodle.
I will give you the question paper. If you would want it on Moodle, I can put it on Moodle. But basically, you will have to write it down. So you'll have to write it down. उसका photo निकाल के मुझे भेजना पड़ेगा. You'll have a certain time within which you will have to take the photographs and send it to me. And because this is a minor exam, if anybody forgets to send me the answer script on time, or if there is a delay, then it is really up to you and not on me. सो इंश्योर दैट अगर मैंने कहा है 1.5 घंटे के अंदर वो मेरे पास पहुंचना चाहिए, तो 1.5 घंटे के अंदर ही पहुंचे. ऐसा नहीं कि अगले दिन आपको लगा कि आपके आउटबॉक्स में रह गया है और मेरे तक पहुंचा ही नहीं है. Because the moment I receive something, I usually send back a mail saying received. If you have not seen a mail that says received, that means I have not received your answer scripts. So be very careful about it. Last time somebody did it, I didn't say anything because anyway that wasn't even an exam. But when it is an exam, be very careful. You have to send it within the stipulated time. अगर 1.5 घंटे का एग्जाम है, 1.5 घंटे के अंदर ही भेजना है आपको. You
So,
write it,
ma'am, it will be
and then you
totally
take a
experimental
photograph
based.
and you send it to me. That's it. Or you scan and send it to me. Or if you're
Ma'am,
typing
I'm
your
saying it.
answers, type and send it to me, whatever it is.
So, ma'am, it will be totally experimental based, right?
I'm not saying it's totally experimental based. What I'm saying is that it's an open book. There, you might be asked to interpret certain results. You might be asked to design experiments, something like that. but it will be open book. So definitely I'm not going to ask you कि एक्टिन में एटीपी बाइंड करता है या जीटीपी बाइंड करता है, वो तो नहीं पूछ सकते ना ओपन बुक में.
Yes, ma'am.
All right, we'll meet on Tuesday, okay? The lecture notes are already on Impartus and you can find them in the backpack. Thank you.
Thank you, ma'am.
Thank you.